So now we're looking at non-Mendelian genetics. Mendelian genetics are such that alleles are either dominant or recessive. But you can have multiple alleles that code for the same thing. You can have co-dominance. So two dominant alleles are two alleles that are equally dominant, both traits show, or incomplete dominance, where you just get a kind of part-way mixture. So I'll give you an example of multiple alleles and co-dominance with blood type. So blood type can be A, AB, or O. I'm not going to get into the negative and positive ones. We're just going to look at these particular alleles. So the way it's expressed for allele type is with an I and a capital A, that equals type A, and it is dominant. An I with a capital B is type B, also dominant. A little I equals O type, and it's recessive. So the combinations that you can get, excuse me, of these are, you could get, because there's multiple alleles, 3. You could get these two together. That will give you type A. You could get these two together. That will give you type A. You could get these two together. That will give you type AB. You could get these together. That would be type B. You could get these together. That would also be type B. Or you could get these together, and that would be type O. So A, B, B, sorry, A, B, 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 and O. And the reason that they can be co-dominant, the A and the B, is that if you have type A, that means that your cells have particular type A receptors that identify your cell. If you have type B, you have a different kind of receptor. And if you have type O, you have no receptors. So if you are homozygous A, then you'll have all these kinds of receptors. And in this case, you'll also have only the A receptors. But in this case, you will have both kinds of receptors. They're not really receptors, they're more like markers. With B, you would have only B. It's supposed to be a square. With B and I, you will also only get the B receptors, because with I, you don't get any. And with a double I, you will get no receptors or no markers. So these markers are what basically label your cells. And so if you were to get a blood type injected into you that was different, then your immune system wouldn't recognize it and it would attack it. So which of these then is a universal donor? Which blood type could be a universal donor? That no immune system will attack. Oh, yeah. So this is the universal donor. Which is a universal recipient? which individual could get any blood type? AB. Yeah, universal recipient. And the universal recipient has two different alleles, but they are co-dominant, which means that both alleles are expressed and both phenotypes are expressed. So that's a case of multiple alleles and co-dominance. Incomplete dominance, I can't think of a human one off the top of my head, but one that's quite interesting that Mendel found is that he crossed red flowers with white flowers, and he ended up with, I'm sure I have other colors, but he ended up with, let's draw it like that. So this is a red flower, it's completely red. This one is completely white. He crossed them and he got pink. So that was a case of incomplete dominance. Yeah, so dominance doesn't mean better or worse or anything like that. It just means that if you have the dominant allele, you will express normally a dominant phenotype. Polygenic inheritance is when there are more than one gene that give you the phenotype. So two or more genes that combine to produce a single phenotypic trait. So skin and eye color is like that. So eye color, for example, there is a brown allele, a blue allele, and a green allele, and they are on different chromosomes. And skin color has 3 alleles, but interestingly with skin color, it doesn't matter which alleles are dominant or recessive, but if you have all dominant alleles of these 3 types, then you will have light skin color. If you have all recessive alleles of dark skin color, might be the other way around, I'm not sure. But if you have all heterozygous alleles for all 3, then you'll have a medium skin color. So there isn't a cut and dried one or the other when it comes to skin color. It's a spectrum. Pleiotropy is a situation where one gene gives rise to many different traits. So sickle cell anemia is one of those. It's only one gene. It's only one gene. And so there's only one difference in amino acids that in hemoglobin that leads to sickle cell anemia. But what occurs is that the red blood cells become very sticky and fragile because with sickle cell anemia, instead of having nice round red ones, you have sickle-shaped squished ones. So this is normal. Normal is capital S. Sickle cell is recessive. So the hemoglobin gets all bunched up and causes the cell to crenate. But it also causes other phenotypic traits like low oxygen levels, anemia, and an enlarged spleen. Sickle cell is a very interesting case because S is normal hemoglobin, little s is sickle cell hemoglobin. So if you have 2 dominant alleles, you'll have normal hemoglobin and normal red blood cells. if you have both recessive alleles, you will have the disease, sickle cell anemia. And if you have both, if you're heterozygous, you will have normal and sickle cell blood cells. Well, sickle cell anemia is quite a serious thing, and it can lead to fatality before reproduction. So you would think that eventually that allele would disappear from the population, but it does not, because strangely, in some situations, this condition of being heterozygous leads to an advantage. It's called the heterozygous advantage. Which is where a person that is heterozygous has somewhat of protection against malaria. This is a disease that happens mostly in Africa, where there are mosquitoes that carry a protist, a plasmodium, that affects the cells. So it's protection against malaria because these protists that infect cells, they easily infect the large red blood cells, but they really can't affect the sickle cells. So malaria doesn't do well in a heterozygous individual. So these individuals tend to survive malaria, and therefore this recessive gene, which is otherwise deleterious, remains in the population. Genes may also be inherited on the sex chromosomes, and that has some implications. So if you're a female, you are XX, of course. There is an allele for hemophilia. It's recessive, but it's on the X chromosome. So this individual is female and has the other X chromosome with a dominant allele. So this individual will not have hemophilia. A male, on the other hand, that inherits the chromosome with the recessive allele doesn't have, the Y chromosome is much smaller, doesn't have the dominant allele to counteract it. In this case, males tend to have hemophilia more often than females. And a similar situation occurs with color blindness. Color blindness is also recessive, and recessive on the X chromosome. So males are much more likely to exhibit color blindness as a result. Yeah. So our traits are not solely genetic, of course. Our environment does affect our traits as we are adults, and there is a role of the environment. So brown eye color, for example, requires phenylalanine, an amino acid from the diet, to produce melanin, the pigment. So that's the environmental factor. There are two enzymes that convert phenylalanine. So here's an amino acid, phenylalanine. It's converted to tyrosine by one enzyme. So we code in our DNA for that enzyme. Then there's another enzyme that converts tyrosine to melanin. And melanin, if there is a significant amount of melanin, will lead to a brown eye color. So that is the role of the environment. Penetrance is really the percent of the population to express a predicted phenotype given their genotypes. So not everyone does. It's not a simple, straightforward thing necessarily. Sometimes it is, um, but sometimes there are other genes that can affect genes on different chromosomes. So penetrance is the percentage of the population to express a predicted phenotype. So pedigree analysis is something that we're actually not going to do it in this class. I have done it in previous classes, but you do need quite some time to do it. But it is where you predict genotypes given what you know about phenotypes. So we know that these two individuals, this one is a female, this one is a male, expressed the phenotype, in this case, of albinism, which is a recessive allele. Sometimes you can take this information and predict what the genotypes were of the ancestors, and thus what perhaps your genotype is. So pedigree analysis is still used, but more often than not now, we use molecular analysis. So we just take DNA and sequence it. So here's a sickle cell and a normal red blood cell. And that's sort of a misplaced slide. Another trait is known as achondroplasia or dwarfism. And what would you say, just guessing, would you say, do you think it is a dominant allele or a recessive allele that confers achondroplasia? So all of these individuals are expressing achondroplasia at each generation. What would your guess be? Dominant or recessive? So we've got dominant or recessive. Yeah, so at first glance, this might be your just first analysis. You would still have to do further analyses, but dominant would be a good guess because so many individuals have expressed that trait. And we know that the trait will be expressed whether the person is homozygous dominant, heterozygous. So dominance and recessiveness, however, do not determine frequency in the population. Often they are more frequent, but it doesn't mean they necessarily are. Some recessive alleles, strangely, blood type O, are quite dominant. And some dominant alleles, polydactyly, for example, polydactyly is when you have 6 fingers or 6 toes. It's quite rare, but it's a dominant condition. And AB is also rare, even though they're dominant alleles. So the next subject is cancer, but I'm not going to lecture on that. I'm not going to test on that. However, you should read in the textbook the section about cancer at some point. And I'll end there.